can go
I'm
ahead.
audible. Yes. I'll just start, sir.
Please.
Indu ma'am. Hello. Yeah.
Go ahead, ma'am. Go ahead. We will start it.
Yes, sir. Good evening to one and all present here. I once again extend a warm welcome to you all to the second session of today's webinar. I'm happy to introduce the resource person for today's second session, Dr. Sushil Kumar Midda. He is an associate professor in the Department of Biotechnology at Maharani Lakshmi Ammani College for Women. He's a review editor for the
Good evening,
board of
Sushil sir. Are you
Very
there?
good evening, ma'am. Very good evening.
Hello,
Yeah, ma'am, I can hear you.
Dr. Nutana.
Yes, ma'am, I'm here, ma'am. I'm starting the meeting. Hello? Hello, ma'am.
Sahana, can you hear me? Hello.
Indu, ma'am.
Yeah. Yeah, now I can hear you. Yes, Sahana. Yeah, I think my volume was low. Okay.
Shall I start
think-
the meeting, ma'am?
Yes.
Sorry for the disturbance. Good evening to one and all present here. I once again extend a warm welcome to you all to the second session of today's webinar. I'm happy to introduce the resource person for today's second session, Dr. Sushil Kumar Midda. He's an associate professor in the Department of Biotechnology at Maharani Lakshmi Ammani College for Women. He's a review editor for the Board of Developmental Physiology. He has also been associate editor for pharmacognosy research and editor for International Journal of Fundamental and Applied Sciences. His ongoing projects are funded by BICEP and DST. He has published many research papers. In yesterday's session, we learned about various types of reports like PRISMA, CONSORT, STROBE, and we learned about the guidelines for writing reports. Now, I request Dr. Sushil Kumar Midda to kindly present his talk on knowing various types of research reports. Over to you, sir.
Yeah, thank you. Thank you very much, Dr. Nutana. It's nice listening to you again. So,
Thank you.
Yesterday, welcome, ma'am. Yesterday, we have started with a few types of reports, if you remember. So, can I share my screen? Is it okay?
Share now.
Yeah, you can see now. Is it okay?
Yes, sir.
Great. So we were discussing about the various type of studies and various type of reports. Okay. So I hope yesterday you were able to follow me. So to start with, I just a little quick review. I will do it. Just a minute. Yeah. Let me do a little quick review. Okay, so let's do it. So we have done with the scientific format and why we need the scientific format and what is the reporting of scientific studies and what kind types are there. So in that we have gone through with the PRISMA, CONSORT, STROBE. I have told you about the only 3 types of studies, though I have discussed here few 11 guidelines. I said 11 guidelines, but I have only taught 3. Hope you are comfortable with it. If anyone has any problem with them, so you can ask me any questions regarding that. Otherwise, I'll start my talk for the day today. I'm very sorry. So a little late, we are starting. I think we are 3 minutes late. So let's start it with them, with the study. So PRISMA, we have studied, CONSORT, STROBE, and then I will give you a little glimpse of formats, prescribed format, like as example, section of the paper, section of the reports type things. Then I told you about the guidelines. Then I said some types of reports. People usually think their scientific report is only the scientific one kind of report. So there are trip reports, progress report, lab report. You have feasibility recommendation reports. So incidents, investigation report, minute of meetings reports, that MOM is called, proposals. This all out of this few, you are going to learn it. You're going to learn soon. This I think your teachers are going to take care of a few things. Okay, so let's touch upon one by one to each and everything. So practice, I told you, ask a peer to review your introduction. So I gave you this question. So please write a report, a small one-page report. I asked you to write it. So I can ask, so we can ask the peers to review your introduction part, discussion, review suggestions, whatever things are available here. So we can do that all. Okay, let's start the studies first, then let's go with the next part of the working styles and whatever we are learning for it. So now I just wanted to tell you about a few more things. We have to start from thank you. So you listen to me. You would be thinking why I'm saying thank you in starting itself. Okay, I will give you a small story. There were scientists, and they were searching few of things. Okay, the story begins here. So in this, the scientist was searching. Okay, discovery they were wanted to do for the. You would have heard about the drug the painful development of penicillin. How it was so much painful. Do you know that? You know that? Who did that penicillin story? Who made that penicillin? It was accidental discovery. You know that? It changed the world. It changed the course of medicines. The people who have never thought of it can be done. And it was not done by the scientist. You know that? It was done by one lab technician. And he was often described as a careless lab technician. Very, very careless. So what did he do? So the story starts with Fleming. He was on 2 weeks vacations. Usually being a student, or like now lockdown is there, it is like closed everything. So you don't know what you kept it back on the positions in microbiology lab or not. I don't know whether you left it your beaker or your Petri plates lying outside and you just ran away. You just ran out. You just said, okay, let us come back. Then we will do this. Then we will do that. Same way, the great Fleming. He oftenly described as a careless lab technician. Very, very careless. He returned from 2-week vacations to his lab and he find a mold. It has developed accidentally and contaminated Staphylococcus culture plates. Whatever culture plates he has put on, he just left them like that only in 1928. Now it's close to 100th, you can say, close to 80th or 82nd 82nd you can say anniversary of it. So it was the first naturally occurring antibiotics drug discovered and used therapeutically. And the guy who was behind it, he is called Sir Alexander Fleming. he is called as Sir Alexander Fleming. He is a Scottish researcher credited with the discovery of penicillin in 1928. Fleming the Great was experimenting with the influenza viruses in his lab at St. Mary's Hospital in London. He was often described as a careless lab technician. He just left everything. He was not finding anything. He was not getting any result. So what did he do? He left everything and he just gone for a 2 weeks vacations. And after he came back, accidentally, he seen his staphylococcus culture was contaminated. And that too with the one mold. Then he started examining that mold. And he seen that mold was stopping the growth of staphylococcus. It was stopping. Then he got an idea. He just published his article in British Journal of Experimental Pathology. And what did he say? He just wrote few lines. Yesterday I was telling you why observation books are very, very important. It's important because of this. Now listen that lines. The Staphylococcus colonies became transparent. He was also not sure. He just said, and then I told you, you are showing your results. He said his results, Staphylococcus colonies became transparent and obviously undergoing lysis. Obviously, it has undergone lysis. That's the reason something is there in that mold that grown at room temperature for 1 to 2 weeks and acquired marked inhibitors, bactericidal, bacteriolytic properties to many of more common pathogenic bacteria. You can understand that. Fleming described the colony as fluffy white mass, which increases in size and after a few days. So penicillin had no effect against gram-negative organism, but was effective against gram-positive bacteria. This report published by Fleming say sometimes find what one is not looking for. When I wrote up just often at dawn on September, I certainly did not plan to revolutionize the medicines by discovering the world's first antibiotic, a bacteria killer. That's exactly what I did. That's the story over. We are done with the story. We are done with the story. Why did I tell you about this story now? What's the reason? I want to listen from you now. If you tell me what did I teach within 2 minutes, within 2 lines or 3 lines, everyone, I'll understand you got it, my point, what I want to say. Please understand, every studies is not accidental. Maybe the Fleming was very, very lucky. Hope you are not done carelessly, left everything in your lab and came out, because it might go for hazardous also. You don't know what can happen. So now let's hear from you. Before I start my talk, another 10 minutes I want to spend with you guys and want to listen to you all. Just now it's 5:49. Let's start with few. Yes, it's thank you slide is showing because I am just presenting myself here. I am showing Sir Alexander Fleming working in his lab. That's why thank you slide is my real slide is next one. So you guys can talk to me. Let's do it. Interactive session, not like regular classes. Let's play around. I got a hand from Sarojini. Sarojini, could you please start? Okay, could you please unmute yourself and say within 2 lines? Sarojini? Sarojini Kadam. She has raised the hand. Divyashri also raised the hand. Divyashri N.
Yes, sir.
Could you please go ahead?
Yeah. You just taught us this.
First line, first thing to start with. One minute, Divyashri. First thing to start with.
Which led to the discovery of penicillin. I didn't get you, sir.
Oh, 2 minutes, Divyashri. Divyashri, don't say you taught us. You make your story and tell me, okay? You are the scientist working in Fleming's lab, or you are scientists working with us here. We all are peers now. In scientific language, it's called peers. We all are peers here. PEER means we all are colleagues here. We all are researchers here who are gathered here to gain knowledge from each other. Okay, Divyashri, let's start. Yeah, please start.
Okay, sir. Okay, the accidental situation which led to the discovery of penicillin, that is when Alexander Fleming left the plates, the bacterial Streptococcus species plates for 2 to 3 weeks. The mold which had grown on it had some effect on the bacteria, that is bactericidal. Yeah. That's all, sir.
That's lovely. That's great, Divyashri. Now, could you interpret the result here? Could you interpret the result? Could you tell me in 1 line? If you want to say something in your lab, you want to write in a research paper, what do you write? In a single line. Okay, let me make your life very easy. I will tell you what I would have written. Some of the other compound present in the mold responsible for the bioactivity and hindering the growth of Staphylococcus. That's it. That's it. That's my interpretation. My result, my research is over. Now, the future generation, what would I give the future concept? I'll give a future concept to everyone stating, everyone, if the future is still open to find out that compound which is hindering the growth of that bacteria. Done. That's it. Sarojini. Yeah, Sarojini Kalambi. Go ahead, please. Miss Sarojini. Miss Sarojini. Go ahead, please, Sarojini. Soumya Shankar. Could you please go ahead?
Good evening, sir.
Very good evening.
Yeah, so he would be Alexander Fleming would be guiding the future generations.
Soumya, 1 minute, please. Say good evening, peers.
Good evening, peers and friends.
Yeah, now start.
Yes, thank you. So Alexander Fleming would be indicating future generations to see that the particular substance that is lysing the bacteria there would have developed probably in this time frame, because he has left it for 2 weeks. One indication, he might be guiding the future generations towards that, and how it is lysing it. How did the action? How did the pathophysiology proceed? He's giving an indication for the further generations to see the pathophysiology of how it is doing it, and what are the things that are causing the lysis. How can we develop that? All these things he's indicating towards by giving that particular information to us.
That's great. That's great. Still, we are left out with 5 more minutes. If anyone else wants to say something, can speak.
I would like to add another thing, sir.
Please go ahead.
Though it was accidental, in one of the morning sessions, one of the doctors did mention that any discovery, it might be negative, but it is a discovery. So even if we don't get the results that we wanted to in our research, if we get any result, that will indicate to something that will be useful for some other study. So it is still a positive thing for scientific background, for the scientific discoveries.
There are no negative. There are no negative things. Correct. This is I don't think so. Soumya, it is negative research. It's accidental research. You cannot say negative. It's accidental research, though it leads us to the first discovery of antibiotic, penicillin. So it's a positive research only. See, every research is positive, negative or positive.
Yes, that's what I meant.
All the researches are positive. Don't worry. That thinking matters. How do you take this type of research? Anyway, let's go, and we have done with the time. So let's start with our class now. Okay. Thank you so much. Thank you. Others. So let's start with our class now. So yesterday we have finished with 3 types. Now 4th one we are going to start with, called STARD. STARD is Standards for Reporting of Diagnostic Accuracy Studies. Standards for the reporting of diagnostic accuracy studies. Okay, standards for the reporting of diagnostic accuracy study. This has been developed for accurate reporting of diagnostic and prognostic studies. Has been developed for accurate reporting of diagnostic and prognostic study. A survey about the diagnostic studies published in Medical Journal 1978 to 1993. Okay, the quality would be very, very poor. So they were not having any study design. They were lacking that. They were not having proper methods to display, and difficult to evaluating their results. Thereafter, STATS came forth with a checklist of 25 items under the following headlines. It's called title, abstracts, keywords, introduction, methods, results, discussions, then some more subheadings are included. So flow diagrams, always a recommendation to report the methods used for the patient recruitment in the order in which the tests are carried out. Okay, so hope you would have understood the STAT things, whatever I wanted to discuss with you. Please remember this all, whatever data I am showing here, whatever things I am showing here, it's not mine. It's collective measure. It's a collective things taken from here and there. So ethically, I should say I have taken this all things from the PowerShow. One PowerPoint presentation things are there. That's called PowerShow. From there, I have taken this all data I have collected from here and there, and then one presentation is called PowerShow. So you can also check that, and you can get this type of data from there. So it's very good website. Okay, powershow.com. So next, I think I have to go for number 5, spirit, SPRIT. Okay, SPIRIT, spirit. So spirit is standard protocol items. I is items. Then R is recommendations for interventional trials. Interventional trials means it was created 2007 for scientific trial protocols. This is the time the clinical research industries came in the boom in the market. It has become after 2005 to 2010, 2009. Whoever has tried, they all received. You can understand, they all got in. But if you have in you some of the other quality, then only you got in. Okay, it's not, I should not say, but whoever people have tried, they all got in, and they all started, they all got trained in the clinical research industries. They all are now at very good stages. So for that time was the golden time for the clinical research industry in 2009. It is okay. For the reporting of scientific trial protocol, as many of them lack the information of primary outcome, treatment, allocation methods, use of blinding studies in randomized trials. So it includes 33 items. Again, it divided, but here they have added administrative information also. In previous reports, you would have seen there was no administrative information was available, but here you got it administrative information also. You got then introduction, methods, and then ethics and disseminations and appendices. Here they have added extra things, administrative information, appendices. It was recommended, okay, it recommends a protocol in specific format that includes table of content, section headings, glossaries, list of references. We are going to discuss list of references in big way. Okay, we may discuss, we may learn Mandala software also. I may teach you a specific software called Mandala. For that, I need a small help from you guys. If you have a laptop with you or system available with you, even mobile, whatever things are available with you, kindly download Mandala, Mandely, Mandala software in your systems. So it would become easy for me if you guys do it with me itself. It will be great help from your side. Okay, I don't want students, I want peers who can work with me here. Okay, so let's work out in that way. Hope you are listening to me properly. Okay, so next type is called CARE, C-A-R-E, CARE, consensus based clinical case reporting. You can understand that this is the today's world going on. This type of reporting is going on. It is more transparent, it is more accurate, and usually it is used by doctors in case reporting. CARE, consensus based clinical case reporting. Next is called SRQR. SRQR, standards for reporting qualitative research. Standards for reporting qualitative research. This is formulations defining a standards for reporting qualitative research and preserve the requisite flexibility to accommodate various paradigms, approaches, methods used. So you could understood here, you would be understanding, you would be seeing it here, the standards what we follow up here. We consisting of 21 items. Here 21 items are there. Okay, and in this SRQR is the major part what they do it. They provide examples from recently published articles. For example, whatever you give that report, you are going to verify with the recently published articles. That's the specialty of SRQR. SRQR. Usually, if you go to pharma companies, they do follow concert type of reporting. Concert type of reporting. If you go to pharma companies, consolidated standards of reporting trials. So this is the major concert type people usually do it in pharmaceutical companies. Okay, so next kind is called SQIRE. SQUIRE. So SQUIRE is standards for quality improvement. Standards for quality improvement, reporting excellence. Reporting excellence. Standards for quality improvement, reporting excellence. Guideline provides a framework to report any new studies conducted for improving the healthcare communities. So this is guideline. They provide the guideline, SQUIRE. Please remember this all wording. Next comes cheers. It's a consolidated reports. Consolidated reports on health, economics, evaluation, reporting standards. Why do we need this type of report? Because it will help to consolidate the health economics evaluations. So evaluations happens with the economics year. Economics matters a lot. How much economy will loss happen because of Corona? How much economic burden has come to a state or a country because of a epidemic. So you can understand this is an attempt to consolidate and update previous health economic valuation guidelines into the useful reporting guidances. You can understand that. It's very, very user friendly. It's always has some same type of things are there. 24 items it uses in the checklist and divides in titles, abstract, introduction, methods, results, discussion, and other information. Next comes arrive. Usually arrive is used for in vivo experiments. So animal research, reporting of in vivo experiments, it's called arrive. So any area of bioscience research that uses laboratory animals to quality of the information, to improve the quality of informations, publishing, and reduce the number of animal studies. There are different types of guidelines available. As example, OECD guideline. These all guidelines are available. Whenever you follow them, you can work out on the animal research. To come back with animal research, reporting of in vivo experiments. For that, you need to have CPSC guidelines, IEAC guidelines, whatever guidelines it follows, all the things need to be written. Note the things from where you got information, from where you started working on, and reduce the number of animal studies. Agree reporting checklist, AZREE. Okay, agree reporting checklist. It needs in practicing guidelines developers, guidelines for users, funders, peer reviewers, journal editors to improve the comprehension, completeness, and transparency of the reporting and practicing guideline. So here, all the editors, all reviewers, all developers sit and take a concurrent decisions how to evaluate the reports. Whatever we are writing, how to work out on that. What reports can be done, how reports can be used. So this all can be done. Now we come down to the agree. The purpose of having these reporting guidelines in scientific research is to create a manual for the authors to follow and promote total transparency, accurate reporting, and easier accessible of the validity of reported research finding. So though it has been accomplished to some degree, but still needs to be done, it's time that scientific editors, authors, journals, reviewers assemble to figure out how the best use the reporting guidelines to. So now hope you would have understood all the guidelines. Now I want you to guys to write down all the guidelines names. I'll give you 2 minutes time. Then we will go and work out on reference patterns. Yeah, someone has any questions? Yeah, if any questions are there, you can ask me first. Yeah, please. Any questions? Yeah. Any question? Okay, no problem. You can look for now. You just write down, take down all the guidelines what I said about, what I just now discussed. I want you to write all the guidelines name, so I will understand what you learned it. Can you jot down all the guidelines? All the 11 guidelines. Then I will tell you about scientific editors, what they follow. They follow COP, COP guideline, Center for Publication Ethics. They follow COP guidelines. So they follow COP guideline. COP is called Committee on Publications Ethics. This promotes all the integrity, whatever we do in the publications. COP, COP guidelines, they have to follow. If you want to see any journal, whether the journal is doing good or not, you can always look for whether they are member of COP. Yes, another 4 minutes I'll give you to fulfill, to write down, take down all the names. So we are going to work on Mandala software. Okay, it's online also available, so nothing to worry about. It's available online also, so do not worry about. Kindly start texting your answers so we can start working on your next part of the talk. Divashri, good. Next person, please. Good, Hemavati. Next person. Ayushi, we have not started Mandala till now. Prism, Sushmita, Prism, it's not Prism, it's Prisma. People started copying from each other or they know it. Good. So we are doing well. That's great. Anyone can speak about it, about all the 11, the names of it. Anyone out of you? Sahana, why don't you try? Okay, done. So let's not break our head here now. So let's start with the next session. Literature writing, literature citation, how to write references. Let's start doing with the reference patterns. So have you heard about the reference pattern? Anyone? Do you know what is the reference pattern? What does it mean? I'll give you 2 minutes time to go through, check on Google, just write citations, how to write literature, how to write citations, and just get back to me. 2 minutes. Within 2 minutes. It's 18:18, 6:18. So 2 minutes, 6:20, I will get back to you. I'll come back to you. Go through, just check what is the citation, what is the reference patterns, and then you have to tell me about it. Let's go with that. So let's start with referencing pattern. 3 Sana to everyone. What does it mean, Sana? Sana, what does that mean? 3, write a sentence about the idea. 4, write a reference list entry for the work. What does that mean? Okay, you want to say read the work, you want to cite. Okay, great. Let's go ahead. So let me share my screen. I'm sharing my screen. Okay. So where I am, can I share directly my COOP? So I'll write it. I'll share COOP. Okay. So look into that. So look into that. What is the COOP now? So I'll open COOP. COOP is a Committee on Publication Ethics. Okay. So you look into that. Okay. Now let's start with the Mandala. First, I'll teach you the software, then I'll teach you the reference pattern. So all of you who are new here, you need to download Mandala from here. Okay. You need to download Mandala here. Okay. You need to download Mandala here. After you download the Mandala here, you look into this. If you open this like this, it will come. Okay. So here you guys need to download Mandala here. After you download the Mandala here, so depends on your system, which type of 62 for 64-bit or 32-bit you download here. Accept all the cookies. After that, it's for Linux. So for that, now you have to see for Windows. So just have a look for here for Windows. Okay. If you click on this, it's download for the Windows. So you can download it. After you download it, you should like it starts getting downloaded. After it starts getting downloaded, you have to open it. I already have the Mandala. After you open this one and then run it, after running, it will come like this. Oh, I have to stop it. 1 minute. I have to pause it. Anyway, it happened now. No problem. It happened. Okay. So we have Mandala here. So like this, it will happen. It will come. So you can see this. Then so you download it. All of you have downloaded now. Could you please do that? Could all of you please do it? It's okay, Sachiri. Now please download the Mandala software. Are you able to do it? Kindly put thumbs up in the chat box, whoever has done it. Otherwise, I'll show you demo. That's the last sources. If you don't go with me, I will do the demo. So if you're done the downloading Mandala, let me know. Or you say, I will do the demo. Shall I do one demo? Just write down in the chat box. Thank you, Sana. Others also please start writing. Others also please write. Okay, then I'll do a demo today. You guys, what you do, you practice it afterward. Anyway, next class again, I am going to work out on with you. Then we can do the Mandala things. Is it fine? Hope that would be fine with everyone. So let's start with Mandala. Okay. So I'm sharing my screen. So I'll share my screen. Okay. I'll share my screen. Can I share multiple window? Can you see my screen? Just let me know what you are seeing it. I'll open Mandala. Okay, I'll open Word file. I don't know what is there now. Okay.
But nothing is visible, sir.
Okay. I'm sorry. I'm trying that only I'm not able to. Share is automatically getting disconnected. 1 minute. I'll do this again. Could you see? Okay, I'll just share my Word first. Can you see my Word file?
Yes, sir.
Okay.
Word
So-
file is visible.
Okay.
Yes.
So I'll just start typing something. Okay. I will have some research paper. Okay. So I'll say diabetes is multifaceted disease. Okay, and then I will write about few more lines. Okay, I think I'm going to write about some more things like about the some more projects files. So oh, I'm sorry, it was muted. So you can see one document has been made. Something has been written in this, so do not worry about it. So now look at this, the document. So what document says? So first way, when you're writing the paper, when you're writing a report, you should have a same type of typing. See the typing style is the MCQ number 11. If I said 12, if I want to have a gap of 2, so everything should be 2. Okay, if it comes under introduction, okay, if it comes under introduction, it's great. If it comes under introduction, then I can select it, I can make it bold. Okay, it's here, so that we have to follow up everything. Now I want to write, I want to give here a reference. So what do I do? Whenever I finish the Mandala, you have to go for here the references. You can look into the here. Your Mandala start seen like this. Okay, all of you have in Word when you download it. Now, if I click on this Mandala, it will say insert citation. I say Midda. So look at that, automatically my citation starts coming. Okay, I said Midda, or I want to say diabetes. See, on the paper on diabetes started coming on the name of whatever papers are there. So I clicked on this and then I said, okay. I said, okay, and said dot. Look into that. All of you, can you see this number has come? All of you are able to see this? Some numbers start, some numbers start visible. So this is American Medical Association style. If I change it to the IEEE side, look at this. Style is changed. Look at that, style is changed. Now I want to make it VanQR. I want to make it as a national laboratory of medicine. Gurika, could you please mute it? Then Indian Journal of Cancer. Look at that. I can take any style. VanQR. I can take any style. See, this is American Political APSA style, AMA style. So this is the way it can be done. Then next I want to put here some other document that's on kinamate 3-phosphate or schemate phosphate. I will go select it. I'll go here. Wait. I'll go insert citation. I can search it here. I can search it here. Oh, I can search it and I can put it here. So if no schemate is there, think pathway. I want to put some pathway. So I put it here. I say, okay, it has come here. Okay. So down here, I want to put now some citation. This I am putting randomly. Please don't do it. I will tell you how to do your citation. You have to bulk up your citation. No one asked me from where these citations are coming. It's not blindly coming. It has been already created. It already made. Okay. So now I want to put it for amino acid. This is amino acid. Say okay. Now look at this. Could you see citation? Now if I change as I want it, if I see Divya, now it has come. Now down, now the major part comes. I say insert bibliography. Look at that, it has come automatically. References. Automatically it has come. And in American political APA style. Now I want to make it IEEE set. Look at that. Your number has been changed here automatically. Software has done the job. And here also A123 has happened. Could you see the difference here, major difference? I'll show you one great difference here. If I change to American Medical Association, Goel G is coming first. Then M is coming, D is coming, as per the numbers. If I say Chicago, look at that. When Goel et al has written here Goel et al ABCD pattern it has come. See, Divya, Goel, then M, A, B, C, D, E, F, G, H, I, J. Okay. If I change it to the Political Science Association, if I change it to the another numbering pattern, see, numbering pattern means it's not following your ABCD. It's not following, isn't it? So there is difference. So what is happening here? The software is automatically doing it. Software is automatically doing it, isn't it? Correct? So you can understand the way it's happening. Okay. So it's automatically changing. So you are free. Why this software now? Why this software now? See now, it's a Chicago National NLM. See here, I'll tell you the change here. How many authors are there? 1, 2, 3, 4, 5, 6 at all. After 6, it has gone for at all. Now I'll show you with the other style. See here. Here at all. So you can understand every pattern it's follow. See, all has come 1, 2, 3, 4, 5, 6. This all pattern has come. How to create this pattern manually? I'm going to teach you next class. Okay, so you can look into that now. I know it's late becoming. I'm very sorry. Okay, so now look into this now style. It's automatically creating the style. Please remember, if more than 2 authors are there, we give at all, and we follow the names. As example, Sushil Kumar Midda. So Midda at all. We will take the surname. Okay. Tarambedu Usha at all. So whatever name comes as your surname, that will be given as a main at all. The first one. If there are 2 only, what will happen? Sujetna and Jangbu. That's it. Look into that. Up. See. Mezkovic and Jangbu. See here, there's no comma, that's why it's thinking, software is thinking this is the one. So that's the reason it's coming that way. So you can always understand. Now, see now, it's coming as a Divya Shambhugam Chinika. Okay, I'll change it now. I'll change another style. How it's become, see now, look at that, the style. If I change it like this, Goyal AK, Midda SK, Usha T, full name is not there. Magnozik S, Divya S full name has become your permanently surname with that. So then after that, the title, then you have the journal name, then year, semicolon, 7-2. Same thing followed down also. Same thing is followed down also. 2018, semicolon, this colon, page number. So this is very tedious task. I am playing only with the 3. Okay, if you have to play with the 300, then it will become little task, tough task. See, American psychology, in that DOI number is also appearing automatically. It's appearing DOI number also. Reference pattern making is a very tedious task when you write any research paper, any report. And that too, it should be formatted in same way. Look at that now. General biomedical and structure dynamics, 3 biotech. See, all are italic now. Food and function. Think this all you have to do manually. And I'll tell you, in my thesis, I did manually. So you can understand the task involved, time involved in it. So this is the way Mandala works. Okay. Now, if I delete this one, what will happen? If I delete Divya, what will happen? Look at that. You have to click. You have to click again. Look at that. Look at that. Divya is automatically gone. Sorry, I have clicked 2 times. See, Divya is automatically removed. It's automatically gone. It's automatically detecting it. the software is automatically detecting it. So this is the power of Mandala. Mandala is a tool from elsewhere. Scopus. It's a Scopus base. It takes Scopus is again another database. Okay, Mandala is a tool. So don't get confused, Scopus and all of them. Okay, so Mandala is a tool, always remember. So it's a reference patterning tool. Like same type, you have reference manager, you have EndNote. EndNote is not free, it's a paid app. It's a paid software. EndNote, Mandala is free. All of you now have to practice Mandala tomorrow whenever you come. Okay, so next class, whenever I am going to take, I will teach you Mandala if you're not learned it, referencing and reference. Next class will be exclusively only on how to write references, how to use Mandala, and whatever doubts you have. You guys downloaded Mandala. You should practice Mandala today. If whatever doubt you have to come back to me and tell me how to do it, what doubt you have. For that, I will just show you something else also. 1 minute. One problem what you face it is, where is the Mandala? Can you see? Mandala looks like this. When you open that Mandala desktop download, you have to sign in. After you sign in, it will start appearing with your name. Okay. Then on Tuesday, December 25th, January 2022, from 7 p.m., you may be unable to access all the services due to planned maintenance. Okay. So good. Let's try within 1 or 2 days before 25th. Okay. So you can understand this. So they do plant maintenance, then automatically, then you can use it after 1 or 2 days. Now, how to add the data here? You have any reference files, you can add it manually also. Otherwise, you can add any document recently added. You can create groups. You can make like COVID papers all are here now. See, I have loaded all the COVID paper. So it's here. So we can, my publication, it's only Meta et al. Publication. All the Meta's publication, whatever is there, all has come. So you can always do it. Okay. This all you have to try out. If you don't learn it, it won't work out, then let me know. Okay. You can import it from BibTeX and not RIS file also. Okay, you can even export also. Okay, you can do this all. You can synchronize your libraries, whatever libraries are there, you can always synchronize. So I work on pomegranate, so pomegranate data is there. See, it's synchronizing now. You can understand that. So genome paper, whatever genome files are there. Okay, so this is the publications. Now even you can add the new papers. You can see here the keywords, even the all that. I'll just show you. See, these are keywords here. One DPPH, proteins, abstract ABTS, actinomycorrhizal, ADMET. So these are the papers are available here. So you can search always all the papers. Okay, this will automatically take the papers from internet. You can always add it here. You can synchronize. You can open it there. Okay, so you can view it. Okay, library and citation. See, citation styles are available. Think some citations are not available, which you want it in general format. You can abbreviate it here. You can put in more citation. These all citations are available. This is update available. Okay, so you can update it. If you say use this style, your Word file, this style will be available. So this all style, whatever you update available is written, those all I have used it there. So you can understand that. Even you can get more styles here, abbreviations. 3 vertex looks like this. So advances biomedical research. So your all the data comes here. This is called NLM library, National Library of Medicines. Okay, there's one citation style library is there. So these all things you can see it here. So please go through. Okay, go through once, then we will work out on it. We will try working on this. Okay, hope this is fine for you all. If any questions are there, you can ask me. How to search a journal, which journal is good, bad, there is nothing like that. If it is, then that also, if you want me to do it, I will try teaching you those things also. How to search a journal, which journal you feel is useful for your future purposes, and all the things we can always try out. Okay, so even you can manually enter here. Look at this. You can always enter here manually also. Author name like Meta SK. So it starts coming like this. Okay, with the organizations. So it automatically comes. Journal name. This journal year thing 2022. Okay, volume thing 28. 18 18 issue number 4. Pages. Some page. Then abstract tags. If you say save, it will save it. Okay, or you can always do with the IDs also. Some ID you can with DOI number, if you give, that's called date of issue number, or URL number, it will work out. Okay, anyway, so guys, I think it's a long day. I know that you all must be tired. Hope you would have understood now mainly. Just practice it. If you don't, we will do it again. Don't worry about it. Okay, that's so all of you need to download and start working on it. Thank you very much. Any questions? I'm ready to take now. So any questions? I wish all of you understood the Mandala. Any questions? Please put it in chat box. I'm waiting for you guys.
Sushi, it was really excellent. I was really amazed to know so many things. Can you give them some practice session, so that they will do it?
Yeah, next is the practice session only, ma'am. So I told them today to download the software because they would be facing some problem. Like already someone is telling it's not coming on Word after downloading. You have to shut down your system and on it again, it will start appearing.
Okay.
So like that. So we are going to do one practice session for Mandala also. So this is just to, because they asked me to do the first demo. So that's why I started.
Okay, what I feel is it is an open thought. You can also think about it. Even any like-minded people in this team can come forward. We can have one research club kind of a thing, maybe publication club kind of a thing. And whoever needs any help, we can help each other, and we can help students to publish it. And we can even go forward in this. See, there are many people who are doing it, but they're all for payment and other things. Here, instead of money, we can think that you can take authorship, or you can help them in whoever it is. For example, if it is your field, you can help them either in conducting some experimentation or to do the statistical analysis or to do some literature survey or each other. We can help each other. We can come out with one membership kind of a thing at the end, a club membership kind. Those who are serious will get into this job and we can help each other and try to build up the thing.
Yes, ma'am. We can go ahead. Ma'am. No problem. You just do it, ma'am. You just let me know. I will be always there. I can.
Instead of an association, instead of something like that, we can make it like a club kind of a thing, where
True.
it will be only research activities, is only publication activities, not even research activities. We can do publication activities. And I will try to take one session on grant proposal writing and how to secure funds. That session also I will try to incorporate in this whole program.
That's
And
great.
so that we can help them out in writing grant proposals. And in this particular team, we can post only information regarding any invitation for paper publications or chapter publications or any new grant proposal invitations. That kind of a thing we can do it. And same group, we can have one on Facebook and one on, and we can have our Twitter handle to follow this thing. What do you say? If we can go ahead so that we can exchange our things. Any ideas? Anybody who is interested in this can come forward and you can give your ideas also.
Yes, sure. True. We can go ahead, ma'am. We can. You can just put forth whatever things are there, and these students, if who are interested, we can go with the club kind of activity. It would be great idea actually.
Yeah, someone is asking to unmute them. Please unmute whoever is handling, whoever is the host, unmute.
And now it is done, ma'am.
Okay. See, anybody wants to speak, you can speak, sir. Students, participants, and anyone has got any ideas to add to the view, whatever I have projected, so that it can be kept alive. Maybe once in a week, we can all have a informal discussion kind of a thing, and it can go on.
Hello, sir.
Yes. Yeah.
Hello. Myself, Dr. Aziz from Bangalore, National Institute of Medicine. Sir, how we can set the web reference through Mendeley?
Sir, can you come back, sir? I did not understand. Voice is not clear. It's like
how we can set the web reference. Suppose I want to go to the web reference.
You can.
Going to take reference from website.
That also can be done, sir. No problem. That we will do the practical session again, sir. As ma'am told, let's do the hands-on. We can do that also. Today we learned desktop. We will do web referencing also.
Okay, sir. Okay.
We will do it. No problem, sir. I hope today you are able to follow me.
Yeah.
So how did you find Mendeley?
Yes. First of all, we have to download and install the Mendeley. I have already done, and I have created many papers and set the references through Mendeley last 3 or 4 months. But sometimes we face a problem.
We will do it. No problem.
Set the web reference.
No problem, sir. We will do it. I will get back to you. We are going to do hands-on. That time you can ask me these all questions also. We will do with the web. Okay.
Okay. Thank you.
It's a great question. Thank you very much, sir.
Okay. Thank you.
Yeah. Sana says, can you give manual of Mendeley? Okay, manual. I have not made it. We'll try making it. Though help files are already available in Mendeley, so you can just look for that. It's always better if you look for help file instead of manuals, and manuals are available only.
No, otherwise you can, a simple thing, you can make a flow chart for a beginner. A beginner, step-by-step flow chart. If we can do, they will explore it later.
Yeah, I will do it and send
that may be simple.
Great. That can be done. That flow chart I will make it. I will make a flow chart and give you all. No problem. That's a great way. Good idea, actually. Okay, manuals are available, but we will do the flow chart way, and I'll give you. Okay. So it's like good, like I'm finding 191 now. Now only participants started coming down. It's like great, like people
Actually,
are,
morning session, we had 254 participants, Sushil.
that's good, very good.
We had very good participation. I think I'm prompting in my broadcast group. This evening session I didn't post it. I thought all of them are there in WhatsApp group and they will be joining. If anyone who has not joined the WhatsApp group, please join the WhatsApp group. The link I request host to post the link again so that anybody who could not able to join, they can join the WhatsApp group so that you will get updated information there.
But then also in this evening time, 190 people, great, good number. Students are learning well. Great. They are really interested.
And there are many HODs and many of my from my contacts. I have one broadcast group in that I have posted it. So
Sure.
many people have come forward and they in turn informed to their students. But I didn't prompted in this evening session. Otherwise,
No
we would have
problem.
had more than 254. Let me see, but they missed it.
Good to
Please,
listen, ma'am. Good to hear
that link for WhatsApp group is being posted again in the chat box. All the participants, please join through this link if you have not joined earlier. Otherwise, you send this link to your friends who wants to join this WhatsApp group to have the updates, because we are going to have many more interesting sessions. Tomorrow, we are going to have a session on how to identify a journal. It is by Professor Pullaya. He's a retired dean and like emeritus professor from Sri Krishnadevan University, Anantapur, indeed my guide. He had more than 300 books, publications, and more than 700 to 800 publications he has got. He is a voracious publisher. He does many things. I think he can give you some clues and help you out the practical difficulties in identifying the journal and publication. Please tell to your friends, as this is a free session, you people can take, I want as many people can take advantage of it, because you're all youngsters, need particular guidance in this particular thing. Try to do that. Morning session was also very good. Crystal was really crystal clear. She made very well. Yeah, you can get the recorded session. We will give on the last day, maybe all the session links can be posted, and we will, and it is in the open YouTube. You can just go to Padma Shri YouTube and you have many sessions like this channel and you can watch this. Anyway, I request Dr. Indu and Sublakshmi, maybe you can consolidate all the recordings of the sessions, day 1, day 2, day 3, day 4, day 5, like that, and then give a name to that and circulate among the participants. In the WhatsApp group also we can post it. Maybe yesterday's link today, today's link tomorrow, like that you can post it. Whoever will join little late, no, they will just go through that and get back to us.
That's good. Yeah, good going, ma'am. That's nice.
Okay, if there are no questions, I welcome you. It is the same link. We welcome you for the tomorrow's session. It is on publications, identification of a journal. And we have planned a wonderful session on IPR also. That will be after this entire publication and all these things. You will be having hands-on in that also. And later you have one session on biostatistics and research. There are many more interesting topics which are coming forward, which are coming on your way. Try to take the advantage and utilize the opportunity. Over to Nutana or over to the organizer, you can wind it off.
Yes, ma'am. Thank you, sir, for delivering us a wonderful talk and interactive session. On behalf of Padma Shri Institute of Management and Sciences, I profusely thank Dr. Sushil Kumar, sir, for delivering a wonderful presentation and having interactive session with us. I'm sure all of us are getting benefited through these classes, sir. I thank our dear principal, ma'am, for giving us this opportunity. Yes, as suggested by ma'am, tomorrow schedule, I'll just repeat once again. Dr. Pulaya sir's first session will be by Dr. Pulaya sir. It's morning 10 a.m. to 11 a.m. Please note the time and we'll all meet you there tomorrow morning session. And the evening session will be by Dr. Shivakumar sir. 5:30 to 7 is the timings.
And that is also a very important session. Dr. Shivkumar sir is going to talk. He is going to deliver two sessions. One is tomorrow evening, and other one day after tomorrow. He is from IBAB, and he gives a very clear explanation about many things. And we requested him to talk about experimental design, which is very important for all the students, including biotechnology, nutrition, because it's very important when you are trying to do any experiment, how to design your experiment, which type of design you need to follow for your experiments. You need to attend this and take a note of it. Thank you, one and all.
Thank you, ma'am.
Thank you very much.
Thank you all.
Thank you, ma'am.
Welcome, sir. You can end.
Yes, ma'am.